Okay. So, to combat stress. So, think about it. Say you're a smoker, right? And you're constantly smoking, and everybody says that smoking is bad or whatever. And it is, okay? It is bad to smoke or to vape. But in your lungs, there's something called a ciliated columnar epithelial cell. And this type of cell is a specialized cell that is supposed to be in your lungs. I think you can find it in other parts of your body, but it is primarily in your lungs, right? And when you start smoking, there's all these chemicals and all these toxins that you're inhaling into your lungs, and the cells in your lungs, this guy can't handle that. So instead, he's like, okay, you know what? I'm not built for this. So he's going to transform, or he's going to do something, and he's going to become multiple simple squamous epithelial cells. Okay? So basically, he is being replaced with them because squamous epithelial cells can divide faster. They're what is on your skin is squamous epithelial cells, which is why, like, when you get a cut or an injury, usually after a few days it starts healing and it goes back to normal because this is squamous epithelial cells. So in your lungs, you start having squamous epithelial cells to kind of, because they can easily regenerate. So they can deal with the toxins of the lungs faster than this guy can. I don't know if that made sense, but like, Google it or something. I feel like that made sense, but yeah. And there is no real physiologic example of this because metaplasia is not a good thing. Okay, because one cell that's made to do something is being released with a certain type of cell. And even though simple squamous epithelial cells can divide very easily, so they can keep up with the toxins that you're smoking, they're not built to do what columnar cells can do. So they can't filter air or whatever it is that these cells do. And because of that, you start slowly losing function in your lungs because these cells, like, sure, they divide quickly and they heal quickly, but they don't know what they're doing. They're not supposed to be in the lungs. This is. So you can see how metaplasia is bad, right? Because this cell is not supposed to be, these cells are not supposed to be there. So a pathologic response is what I just said. Columnar cells in the lungs are replaced with epithelial cells to better deal with the stress of all the smoke and the vaping air that's in your lungs. Lol. I don't know if that made sense, but I'm gonna assume that it did. Okay. Oh, is that supposed to happen? So that's metaplasia. And the last one is dysplasia. Now, dysplasia isn't really it's not really a form of cellular adaptation, which is why it's blue and the rest of them were purple. And that's because dysplasia is just another form of hyperplasia, right? So a cell starts increasing in number. But here's the thing, it doesn't increase unilaterally. I don't know if I use that word correctly, but it doesn't increase the way it does in hyperplasia. Because in hyperplasia, we have one cell becoming many cells, but it's the same cell, right? So one circle cell is going to give other circle cells. In dysplasia, one circle cell can become a square cell, a rectangle cell, that was a square. This is a rectangle. A pentagon, even. Like, we can get crazy with it because it's not good. And I do want to note that dysplasia is not cancer. Even though it sounds very similar to cancer, it is not cancer until it penetrates the basement membrane. And you're probably thinking, well, what's the basement membrane? That's that's ground zero where the cells grow from. So once dysplasia reaches the basement membrane, then it becomes cancer. It becomes malignant. which is deadly. I think when I was studying for the HESI, even though I ended up not taking it, you had to know the difference between something being malignant and something being benign. This is like a side note, by the way. And benign basically means it doesn't do any harm, like there's no issue, whereas malignant means there is an issue. So do with that information what you will. But yeah. So the thing with dysplasia is that if the stimulus is reduced, right? If you stop smoking, because we're still using the smoking example, like it's still true for this one as well. If you stop smoking in the early stages, it can be reversed. Like the dysplasia can be reversed. But if you don't stop and the dysplasia hits the basement membrane, then you're officially cooked. Then it's cancer. Okay, so those are the types of cellular adaptation. Okay, so we have, I made it so small you could barely even see. You have atrophy, you have hypertrophy, you have hyperplasia, you have metaplasia, and then you have dysplasia. But remember, dysplasia is kind of like the odd one out. Like she doesn't really fit in with the rest of the group, even though she low-key does, but she's different, guys. She's not the same. So now we know what happens when a cell tries to adapt. We know this place. We know this pathway. But what about this one? What about when she cannot adapt? And not because she doesn't want to, but because she can't. I keep saying she, so I might as well make her a girl. So I guess she'd be like a really swole girl over here. Yeah, that makes sense. When she cannot adapt, well, she's going to die. But we're going to talk about the processes that lead to this. So the first thing we're going to say is, what type of stress would happen that she would not be able to adapt to? And that's really easy. That's where we get oxygen and ischemic reperfusion injury. But before that, there's like 3 main things that we need to know, which is number 1, hypoxia, number 2, hypoxemia, and number 3, ischemia. Now, hypoxia is lack of oxygen in the tissue. Okay, hypoxemia is lack of oxygen in the blood. Ischemia is lack of blood flow to the tissue. So basically, think about it this way. If it has emia at the end, then it's something about the blood, okay? It's something about the blood. Hypo means little or none. Ox is oxygen. Even though they're like hypo and ox are kind of connected, just pretend that they're not, right? Because when you pronounce it, it's like hypoxemia. So this is lack of oxygen in the blood. Hypoxia is just lack of oxygen in the tissue. I put down tissue, but it could also be cell. And this one could also be cell too. It's not specific to the tissue. It could happen to cells. So hypoxia means there's no oxygen in the tissue, right? Or the cell. Hypoxemia means there's no oxygen in the blood. And ischemia means there's lack of blood flow to the tissue or the cell. So you can have you can have hypoxia without having ischemia. Okay, ischemia just means that this tissue is not getting blood. Okay, and blood is made up of what? It's made up of red blood cells, which are made of hemoglobin, which carries oxygen. So if there's no blood, there's no oxygen. So ischemia, if unchecked, can and will cause hypoxia. Ischemia can and will cause hypoxia if unchecked, but hypoxia does not necessarily cause ischemia. Okay, so if you don't have any blood flowing to the tissue, then there's no oxygen getting to the tissue. And if there's no oxygen getting to the tissue, then we have hypoxia. But if we don't have any oxygen in the tissue, that doesn't necessarily mean that there's no blood flow to the tissue. I really hope that made sense, because I always try to make stuff as simple as I can for myself, and this was just the way that it could work. So ischemia can and most likely will lead to hypoxia, but hypoxia will not lead to ischemia. Now, hypoxemia is basically lack of oxygen in the blood. So hypoxemia is when a cell gets hypoxia without ischemia. Okay, because the cell is getting blood, so it's not ischemia, right? But it's not getting oxygen, so it's hypoxia. That's basically how that works. Okay, so now we know the conditions that lead to this stress. Now we understand this part. Let's talk about here, where she's not able to adapt. So what happens? But before we do that, let's look at the cell, which is this thing. Now, we all know the cell has something called a mitochondria. And the most important thing about the mitochondria, like everybody knows, is that it is the powerhouse of the cell. And that's basically because of something called oxidative phosphorylation. And if you don't remember this from your AP class or from biology, all you need to know about oxidative phosphorylation is that it uses oxygen to break down glucose to get ATP. And it produces about 30 ATPs. You don't really need to know how much ATP it produces, but it's gonna all make sense. So I'd say just keep that number at the back of your head for now, but it's gonna make sense very soon. So the main thing you need to know about oxidative phosphorylation is that oxygen breaks down glucose to produce ATP. There's a bunch of other stuff going on here, like the electron transport chain and everything else, but you don't really need to know that for patho. You just need to know that oxygen breaks down glucose to produce ATP, and it makes 30 of it. You don't need to know how much, but this number is very important, so keep it at the back of your head. Ooh. Okay. Next thing is, so now we know that the mitochondria gives us ATP, but why is ATP so important? Well, here's your list. And this is just a few. This is like the main stuff, but here's the 3 main things that ATP does. Number 1 is it works with the sodium-potassium pump, which pumps sodium out and potassium in. Okay? Just remember that. I feel like I've told you to remember a bunch of things, but trust, they are going to come into play very, very soon. So the sodium-potassium pump pumps sodium out and potassium in. You know, I feel like there's a mnemonic for that where it's 3 sodiums out, 2 potassiums in, and there's like a rhyme, but I don't remember it, so just, yeah. And then ATP also helps with protein building. Remember, ATP is basically energy. So the cell needs energy to do all these things. So it helps in protein building, and it helps with membrane transport, which is just moving things in and out of the cell and across the cell membrane. So let's talk about the sodium-potassium pump. So this diagram over here, this one, this isn't really what a sodium-potassium pump looks like. I feel like it's more circular and it's probably got like a bunch, but you could probably Google it if you're really interested. But that's just like what it looks like. But that's what it's going to look like for now, because I said so. And, okay. So if you can see in my diagram, you'll see there's more sodium outside the cell than inside the cell, and there's more potassium inside the cell than outside the cell. Inside, we only have 1, 2, 3, 4. We have 4 potassiums outside, and K plus is potassium. This is, I think, chemistry knowledge. Na plus is sodium and Ca plus is calcium. Just in case you needed like a refresher on that, that's what that is. So there's more potassium inside the cell than there is outside the cell. This is a normal cell. This is a good cell. Okay, this is what a good cell looks like. We have our sodiums outside, we have our potassiums inside, and we have our calciums outside. But if there's no ATP, right? Let's start from the very beginning. If there's no oxygen due to hypoxia, right? Or due to ischemia, which is lack of blood flow, which indirectly means lack of oxygen, because remember, ischemia can cause hypoxia. If there is no oxygen, well, then there's no oxidative phosphorylation. Okay, let's just cancel them out. If there's no oxygen, then there's no oxidative phosphorylation. And if there's no oxidative phosphorylation, then there's no ATP. And if there's no ATP, then there is no sodium-potassium pump. Okay, and if there's no sodium-potassium pump, what's going to happen? Well, two things. All the sodium that was supposed to be pumped outside is not going to be pumped outside. So it's going to be in the cell. And all this potassium, that was supposed to be pumped inside the cell is not going to be inside. It's going to be outside. So we're going to have all this sodium in the cell and all this potassium out of the cell. And that's not good, right? Because a normal cell has sodium outside and potassium inside. So this cell is not normal. And the cell also has something called a calcium pump, which is responsible for pumping calcium out of the cell. And if there's no ATP, well, then there's no calcium pump either, right? And if there's no calcium pump, then all this calcium is going to be in the cell. And I want you to know that these two things are happening at the same time. Like these two things are happening. Like there's no this drawing is one. I just separated it because it was easier for me to think about it as two different things. But this is the same thing. So it actually looks more like this. Let's make that bigger. Okay, so there's no potassium in the cell. There's all this calcium and sodium in there that's not supposed to be there. I guess in a real world scenario, one of these potassiums would probably still be lingering around in here just because like life happens and shit happens. But for this example, because none of these pumps are working, all the sodium is inside, all the potassium is outside, and all the calcium is inside. And this is what our cell looks like. Okay? Now, water, so when you're cooking food, stay with me here. I feel like you probably think I'm going off topic, but stay with me. When you're cooking food and you add way too much salt, what are you supposed to do? You're supposed to add water, right? Because the water is going to make the food less salty. Because that's something my mom always used to say is if there's too much salt, add water. If there's too much pepper, then add oil. So just remember that if there's too much salt in a cell, what's the body going to do? It's going to add water. It's going to add all this fluid into the cell. Okay, and what happens when that happens? Well, that's really simple. The cell is going to expand and get really, really big. Okay, really, really big. The cell is going to swell. And when the cell swells, it's basically filled up with water. There's just all this fluid floating around in there. And you have to remember, this isn't what the cell looks like, right? Like all the cell parts and organelles are still there, right? Like the nucleus, she's still there. The mitochondria is probably around here somewhere. The endoplasmic reticulum is probably here. And these are just parts of a cell, okay? In case you you've forgotten, these are just parts of a cell that I'm listing off. There's probably like a Golgi apparatus somewhere over here. And all these cells, right, are drowning in water. And when you're drowning, you probably know what it's like to drown. If you've ever been around a pool as a child, it's just really bad. You're not able to think properly, you're panicking, you can't do what you're supposed to do. And that's what's happening to all these cells. They're drowning. They're not able to do the things they're supposed to do. Okay? And because of that, your body, and if they're not able to do the things they're supposed to do, let's take a second before I go into that. Let's take a second and think about what each part of the cell does. The nucleus is responsible for, I think, RNA synthesis or something. Yeah, I think so. The nucleus, and don't forget there's ribosomes in here too, which are also parts of the cell. Okay, this is AP. I think I did this in AP1, I'm not sure. But yeah, there's ribosomes in here too. And they're also drowning. Now, ribosomes are the primary site for protein synthesis. But if they're drowning, well, there's no protein synthesis. So this is going to stop. Remember, this is all happening because we're not producing ATP. Okay? And we're not producing ATP because there's no oxygen. So this is all interconnected. It seems like a lot, but it's really just going step down. So if you have hypoxia because of like that story that Professor Hannenbaum told about how she was in the mountain, there was a lot of pressure up there. And so her lungs weren't expanding and, deflating the way they should. So she wasn't getting enough oxygen. And because of that, she wasn't producing enough ATP. And because she wasn't producing enough ATP, all this bad stuff was happening. And this is where we currently are in the process, right? So the ribosomes aren't working. So there's no protein synthesis going on. And there's no protein synthesis, well then, there's no proteins. Which is really bad, but we'll talk about that later. Right? So this is just one example. So there's no protein synthesis. And if there's no protein synthesis, well, the cell... Okay, actually, there's no protein synthesis. Let's cap it there so it doesn't get too complicated. What else is happening? Well, since the cell is getting bigger, there's going to be all these holes in the membrane. The cell is going to start swelling. Like, the cell membrane is going to start, like, getting bigger and bigger. And eventually, if the cell doesn't get its shit together, the body is going to send out something called a white blood cell. Now, I don't really know what type of white blood cell it's going to send out, but white blood cells are like the police of the body. They basically go and eliminate whatever is not right. So if this cell doesn't get its shit together, this guy is going to come over here and kill it. Or it's going to kill itself. But this cell, if this keeps happening, all right, if there's no ATP and these pumps don't start working, bad things are going to happen. So what's the cell going to do? If it doesn't have oxygen and it can't perform oxidative phosphorylation, it's going to switch to another form, which is called anaerobic glycolysis, which does not require oxygen. Let's see if I can make that a little bigger. Does not require oxygen. So when I was doing the research for this, I found that there's both anaerobic glycolysis and aerobic glycolysis. But when I searched them up, they both gave me the same definition. So I don't know which one is which, but just know that for this process, no oxygen is needed. So instead of oxygen breaking down glucose to form ATP, here, glucose is going to be broken down into pyruvate. Pyruvate is going to get broken down into lactate, and then lactate is going to get broken down into ATP. And guess how many ATPs we're producing? 2. Compare that with the 30 ATP that oxidative phosphorylation was producing. That's why oxygen is so important to the body, because it's producing this much ATP Whereas this other method is only producing 2. And that's not the only downside to anaerobic glycolysis, because to produce ATP, it also has to produce lactate, which will become which is very acidic. And when lactate is acidic, it's going to increase, it's going to do something called metabolic or metabolic, however you want to pronounce it, acidosis, which is bad. Like, you could just Google why is metabolic acidosis bad. It'll give you a bunch of reasons, but this is like not good. This is not what the fans want to see. Okay, this is literally what the fans don't want to see. So the body starts doing this. So you can think about it this way. If it doesn't produce ATP, it's going to lyse, and all this bad stuff is going to happen to it. If it does start doing this method to produce ATP, it's going to start making the body more acidic. And that's not good either. So either way, the cell's kind of cooked, which is why oxygen is so important. And if you want to find a way to connect this to our ABCs that we learned in health assessment, airway and breathing are literally the most important things, because they and even circulation. Like, even you can even find a way to link circulation to this. All these 3 things prioritize getting oxygen to your body, because if you're not getting oxygen, well, then you're kind of cooked, okay? Because this is either this happens or this happens, and we don't want any of those. So that's why we prioritize these 3 things, because 1, your airways needs to be clear for you to get oxygen. 2, you need to be breathing for you to get oxygen. 3, your blood needs to be circulating so that oxygen can go to different parts of your body. That's why this is so important, because if we don't, this is going to happen, okay? Let's move on to the next thing, which is ischemic reperfusion injury. So we already know what ischemia is, right? It's lack of blood flow to the tissue and cell. So think of this as a blood vessel, right? And this thing right here, this thing that I'm moving around, is something known as a thrombus. And a thrombus is basically a bunch of platelets that just kind of stuck together. I don't know why, like, it just is. And it doesn't have to be a thrombus. It could literally be anything. But imagine, let me get rid of this. Cut. Imagine this thrombus got stuck in your blood vessel, okay? And this blood vessel was going to, say, a cell. I'm just going to pretend this is a cell. So this blood vessel was delivering blood to this cell. So when this thrombus gets lodged here, it's going to stop blood flow, which causes ischemia. Okay, that's the ischemia part of reperfusion injury. And we already know that ischemia is really bad. These are all the things that happen. We get lack of blood flow to the tissue, which is ischemia, can cause pulmonary embolisms, heart attacks, organ damage, strokes, oxidative stress. Like, these are all really bad things, okay? And as healthcare professionals to be, clock it, we don't want that to happen. So what are we going to do? We're going to do something known as reperfusion, which is basically when we get rid of the blood clot that's stuck in here. Reperfusion is basically us as healthcare professionals. Now, it could be the doctor who orders some sort of anticoagulant, which is basically a chemical that's going to dissolve the blood clot, or maybe a surgeon went in with some tools and he took it out mechanically, but somehow, some way, this blood clot is getting removed. And you think that's what reperfusion is. So ischemic and reperfusion. So now we know what these 2 things are. And you think this was a good thing, right? Like, we stopped the blood. We stopped bad things from happening. We've stopped ischemia. So now there's all this clean blood that's supposed to be flowing through. But it actually is really bad. Somehow, like it's not good because when the blood clot was lodged in there, all this old blood, like all this blood that was going to the cell that had already gone, is now like stuck here. So it's going to produce something called catabolites, which is basically just a bunch of stuff being stuck in this old blood because now it's filled with cell waste and there's no oxygen here. So there's just a bunch of junk in here. And these junks are known as catabolites, okay? Just so you know. I don't know if that'll actually help, but yeah. So when we reperfuse this injury, which basically means when we get rid of this blood clot, I don't know why I accidentally moved that one. When we get rid of this blood clot, all this new blood is going to meet with the old blood, which has all the bad junk in it. And because this new blood has oxygen, it's oxygen rich and this one doesn't, it has these things. Oxygen is going to interact with these things to produce something known as ROS, which is basically radical oxygen species. Okay? I know it's a lot, but stay with me, which is basically radical oxygen species. And ROS is like really bad. It's really bad because it damages the cell membrane, it disrupts the cell junction, it causes proteins to misfold, it leads to DNA mutations, and it's all these things ultimately lead to an increased cellular permeability. And in an increased cellular permeability, the walls of the cell are destroyed. So every fluid is leaking out of the cell. The good stuff is leaking out of the cell. What is not supposed to be happening is happening. So ROS is like really bad. And because of that, we get injured. So that's ischemic reperfusion injury in a nutshell, right? There's a clot in the blood. We try to remove it. Old blood mixes with new blood, creates this thing. This thing is really bad because it can lead to all of these things, which can lead to cellular permeability. And you remember how I said calcium was supposed to be outside the cell? Because of this, of the membrane being lysed, calcium is going to enter. Wait, no, that doesn't make any sense. Ignore what I just said. Don't think about what I just said. Just remember that ROS leads to an increased cellular permeability, which is really bad because fluid is leaking out of the cell, and it's just bad. This is like an overview. It's connecting the topics. So that's basically, and the cell is going to die. I feel like I should have led with this. The cell is going to die. That's not how you spell die. The cell's going to die. So let's see how that relates to this pathway. So first, the cell's stressed. What's the stress? Lack of oxygen. And because of that, the cell's not going to be able to adapt, right? And because it can't adapt, what are we going to have? We're going to have no ATP. And if we have no ATP, we're going to have increased Na, that's sodium, in the cell. We're going to have decreased potassium in the cell. And we're going to have increased calcium in the cell. And these three things will lead to cell death. Because, okay, maybe I jumped the gun. Let me break it down a little more so you can understand it a little better. But increased sodium in the cell will lead to increased water, because remember, if there's too much salt, you have to add water. And if there's too much sodium in the cell, the body is going to add water. So increased sodium will lead to increased water. Increased water means the cell is going to get bigger. It's going to get way bigger. It's going to swell. Once it swells, the organelles, stop working. And this is actually something, I think it's something called vacuolar degeneration. This was on the slide. She didn't really touch up on it, but this was there. That's basically what is happening. And another thing you should know is that decreased potassium this is decreased potassium in the cell means increased potassium in the outer part of the cell. And this potassium will eventually enter the bloodstream, which will lead to hyperkalemia. And hyperkalemia is really bad because it can cause like dysrhythmias and stuff.